we are going to discuss about plasma membrane. It may be defined as thin, elastic, semi-permeable living membrane that serves as the boundary for the cytoplasm. This is otherwise called as cell membrane or plasma lemma. It serves as the barrier for the flow of components into and out of the cells. Plasma membrane is about 75 Armstrong in thick. Thickness is almost constant in all plant cells, animal cells and bacterial cells. It is formed of glycoproteins and phospholipids. The molecules are arranged in a definite pattern. History of plasma membrane. Plasma membrane was coined by Naegeli in 1855. Chemical composition of plasma membrane. It is formed mainly of lipids and proteins and a small proportion of carbohydrates, nucleic acids, salts and water are also be seen in some membranes. For example, plasma membrane of nerve cells of central nervous system has 79% lipids and 20% proteins, and muscle cells has 35% lipids and 60% proteins. RBC has 40% lipids, 52% proteins and 8% carbohydrates. Different models of plasma membrane. Various models were proposed by different scientists at different periods. They are bimolecular leaflet model, lattice model, trilaminar model, micellar model, fluid mosaic model. Bimolecular leaflet model. Bimolecular leaflet model was proposed by Danieli and Davison in 1934. According to this model, the plasma membrane is formed of two layers of lipid molecules coated with protein. Each lipid molecule has a hydrophobic tail and hydrophilic head. The hydrophilic heads face outwards and hydrophobic tail of the two layers face each other. This model explains the biological and chemical properties of plasma membrane, but not the surface tension. Lattice model. Lattice model was proposed by Ulpers in the year of 1941. According to this model, lipids and proteins are arranged as a lattice or network in the plasma membrane. Proteins form a kind of mechanical framework. The liquid component is distributed in the meshes of the protein frame. Trilaminar model. Trilaminar model was proposed by Robertson in the year of 1950. According to this model, plasma membrane is formed of three layers. Outer protein layer is 20 Armstrong in thick. Middle lipid layer is 35 Armstrong in thick. Inner protein layer is 20 Armstrong in thick. It is also called as unit membrane. According to Robertson, all biological membranes are unit membranes, and this concept is called unit membrane hypothesis. This model explains the lipid content, electrical resistance and permeability of membrane. Micellar model. Micellar model was proposed by Hiller and Hofmann in 1953. According to this model, the molecules in the plasma membrane are arranged in the form of globular subunits called micelles. The lipid micelles are the building blocks of the membrane, and the protein globules are arranged on either side. In a lipid micelle, the lipid molecules are arranged in the form of a circle. Each lipid molecule has a head and a tail. The head is hydrophilic and the tail is hydrophobic. The tail is directed inwards. Fluid mosaic model. Fluid mosaic model was proposed by Singer and Nicholson in the year of 1972. Fluid, which means individual phospholipids and some proteins can move laterally in each layer, and mosaic means the range of proteins resting on the surface or through the phospholipid layer gives it a mosaic appearance. It is the most accepted model because it convincingly explains the transport through the membrane. According to this model, the plasma membrane consists of lipids and proteins. The lipid is in the form of fluid and the proteins are embedded in the lipid in a mosaic pattern. Hence, the name fluid mosaic model. Lipids are arranged in the form of two layers, an outer layer and an inner layer. The protein do not form a layer. Each lipid molecule has a hydrophobic tail and hydrophilic head. The hydrophilic head face outwards and the hydrophobic tail of the two layers face each other. Lipid molecules are not rigidly positioned in their respective places. They are always in a constant motion and undergo lateral diffusion. Membrane proteins. The protein molecules are globular and are of two types, namely peripheral or extrinsic proteins and integral or intrinsic proteins. Peripheral proteins. They are arranged on the surface and loosely bound to the lipids. If intrinsic proteins are removed, the membrane structures are disrupted. Integral proteins. They are deeply embedded and are tightly bound to the lipid molecules. If extrinsic proteins are removed from the membrane, there is no disruption in the membrane structure. The peripheral protein as well as the outer part of integral protein are studded with sugars. Such proteins with attached sugars are called glycoproteins. Similarly, sugars are also attached to the outer surface of some lipids. These lipids attached with the sugars are called glycolipids. The carbohydrates found in intrinsic proteins and glycolipids form a sugary covering called glycocalyx. It recognizes certain foreign proteins as in immune cells and protect the cells from the extracellular digestive fluids. The fluid mosaic model stresses that plasma membrane is semi-fluid in nature. The lipid and the intrinsic proteins move freely within the lipid bilayer. Transport across cell membrane. Transport of materials across the plasma membrane is essential to the life of a cell. It is also essential to maintain equilibrium of cell. Certain substances must move into the cell to support metabolic reactions. Other substances which produced by the cell for export are cellular waste products and must move out of the cells. Types of transport. There are three types of transport found in plasma membrane. They are passive transport, active transport, and bulk transport. Passive transport is further subdivided into simple diffusion and facilitated diffusion. Active transport is subdivided into primary active transport and secondary active transport. Bulk transport is further subdivided into exocytosis and endocytosis. Passive transport. Passive transport occurs along the concentration gradient and without the use of metabolic energy. Simple diffusion. Movement of particles from an area of higher concentration to an area of lower concentration. It does not require energy and membrane transport of proteins. Example, transport of gases and soluble lipid molecules. Facilitated diffusion. Facilitated diffusion is a form of facilitated transport involving the passive movement of molecules along their concentration gradient, guided by the presence of another molecule, usually an integral membrane protein forming a pore or channel. It does not require energy. It is more rapid than simple diffusion. Example, absorption of fructose, glucose transport. Based on direction of movement of molecules, facilitated diffusion can occur by unipode and antipode. Unipode, transport of single type of molecules in one direction. Example, absorption of fructose. Antipode, transport of molecule in opposite direction. Example, chloride bicarbonate exchanger. Active transport. Active transport occurs against the concentration gradient and with the use of metabolic energy. Metabolic energy is used to move ions or molecules against a concentration gradient, which results in the accumulation of solute on one side of membrane. Primary